Students, today we will study another species of Cyanophyceae, that is the Nostoc. It belongs to class Cyanophyceae, order Nostocales, family Nostocaceae. Species of nostoc occur in freshwater as well as in terrestrial habitat. The freshwater species form macroscopic colony on the surface of water which vary from few millimeter to 8 centimeter or more in diameter. Each colony is bounded externally by a pellicle like membrane. Nostoc communae, a terrestrial species of alpine region, form a leathery or rubbery sheath on the damp soil. Some species of nostoc are endophytic and they occur in a symbiotic association with other plants. Example, Nostoc punctiforme is found in the corollite root of the cycas and Anabina thallus. Nostoc sphaerocerum and Nostoc colema are the component of lichen and occur in a symbiotic association with fungi. Some species of nostoc are grown in rice field. They have the capability to fix the atmospheric nitrogen, thus helping in increasing fertility of the soil. In India, nostoc is represented by about 26 species, of which Nostoc calcicola, Nostoc endophyllum, Nostoc epilosporum, and Nostoc mucorum and pulciforme are the common. Thallus, the trichome of nostoc, are uniseriate, usually contorted and twisted in a various way. An important feature is the presence of prominent constriction between the adjacent cell, which gives a characteristic molliform appearance to the trichome. Each trichome is enveloped in a gelatinous sheath, which is a transparent hyaline or colored. Usually, many trichomes aggregate together and their gelatinous envelope dissolves to form colonies of various shape and size. The trichome of nostoc are characterized by the presence of heterocysts, which are intercalary or occasionally terminal, usually solitary, and of much the same size and shape as the vegetative cell. The intercalary heterocysts have the two polar nodules and the terminal one have only one basal nodule. Cell structure. The cell are spherical or rounded and exhibit typical cyanophycean cell structure. The cell wall is of mucopeptide. The protoplasm is differentiated into peripheral chromoplasm and the central centroplasm or nucleoplasm. The chromoplasm has pigment, cyanophycin protein granules, and cyanophycin starch. The central colorless centroplasm is the nuclear region of the cell. Reproduction. Nostoc reproduce only vegetatively. Reproduction, sexual reproduction is absent. The vegetative propagation takes place by the following method. By fragmentation, the colony may break into small fragments due to mechanical, physiological, or other factor. Each fragment has the capacity to develop into a new colony. Second, by hormogonia, the trichome breaks into small segments due to degeneration of the intercalary vegetative cells or because of the presence of the intercalary heterocysts. Multicellular fragments so formed are called hormogonia. They come out of the gelatinous sheath of the colony, grow rapidly and form new colony. Very often the hormogones fail to come out of the parent colony and divide into inside the gelatinous sheath of the parent colony. The result is a large number of trichomes inside the parent colony. Third is by akinete. During unfavorable period, some cell of the trichomes are transformed into resting spores or akinete. They have an additional three-layer coat outside the normal cell wall. The outer layer is variously sculptured. The protoplasm is full of reserve food material. Development of akinete normally takes place in a mature colony. It is always the vegetative cell adjacent to the heterocyst which is the first to metamorphose into akinete. Subsequently, many vegetative cells are transformed into echinate. It is not unusual to find all the between the two successive heterotic transformed into echinates. Metamorphosis of the vegetative cell into echinates is probably stimulated by certain chemical substance secreted by heterosis. The echinate have tremendous resistance for cold and drought. During the favorable condition, these protoplasts become active and break the thick outer wall and form new trichome. By heterosis formation, the fourth method of vegetative reproduction, it is heterosis at the resting spores. The protoplasm of the heterosis of such species become functional and germinate to form a new trichome. At the time of germination, the protoplasm divide by transverse wall to form two cells. Both these cells divide again to form fourth germling. The thick wall of the heterosis ruptured at this stage and the germling come out and develop into new trichome. The fifth method is by endospore. In many species such as Nostoc commune, Nostoc microscopium, the protoplast of the heterosis divide successively to form the endospore. Degeneration of the capsule wall result in the liberation of these round spores which later germinate to give rise to new plant.